Hi, this is Maggie Byers, and this is my presentation on Chapter 12. So, what is the goal of antimicrobial treatment? So, the main idea is that antimicrobial therapy tries to kill or stop the growth of microbes without damaging our own cells. And this is a difficult process because there isn't a perfect drug that can balance effectiveness and safety. So, if there was a perfect drug, it would stay active just long enough to kill the microbe, and it would then dissolve into the body fluids and not cause any resistance. But no drug is able to check all these boxes, so they have to balance all these traits to reach a good medium. So not every antibiotic is technically the same thing. So antimicrobial is a broad term, and antibiotics usually just target bacteria, where antimicrobial targets bacteria, fungi, or viruses. And they are usually described as narrow spectrum or broad spectrum. Narrow spectrum ones are great for target infections, and broad spectrums, they kill a lot of different microbes. So broad spectrum kills many, but they also tend to mess with your normal microbiome. And prophylactic drugs, these are basically a way to prevent infection before the infection even happens. So before antibiotics are given, doctors like to see which drugs the bacteria are sensitive to, and they do this through the Kirby-Bauer test. This test uses paper discs that have been soaked in different antibiotics, and the bigger clear zones mean that the bacteria is more sensitive. And then the MIC, or the minimum inhibitory concentration, this tells us the smallest dose that's required to stop bacterial growth, and these tests help doctors figure out which drugs work and which ones won't. So, the therapeutic index, this compares how much of a drug can cause harm versus how much is needed to be effective. So, with a higher TI, this means you have a larger safety margin, and you can give enough to kill the microbe without hurting the patient and have any side effects. But a low TI is dangerous because the effective dose and the toxic dose are very similar, and doctors will always try to choose a drug with a higher TI if possible. So, selective toxicity. This means that the drug only harms the pathogen and not us. And the best example of this is penicillin. It attacks the peptidoglycan walls in bacterial cells, and this is something that humans don't have. But there are drugs that hit cell membranes or structures that is shared between pathogens and humans, so it can also harm human cells and will result in more side effects. So basically, the more different that the microbe is from us, the more safe it's more safe for us to use. So antibiotics will mess with the vital processes in bacteria. Some stop the cell wall from forming, others block DNA or protein synthesis, some will even punch holes in the membranes, and others will block folic acid production. And folic acid production is what bacteria need to make DNA and proteins. So each drug targets a specific weak spot, and they can also be combined together to be more effective. Okay, even though these drugs are meant to help us, they can still hurt us. So toxicity happens when drugs directly damage organs. So if you ever hear its antibiotic is nephrotoxic, this means it can cause kidney damage, or hepatotoxic, this means it can cause liver damage. And allergies also can occur when your immune system reacts to the drug. Like, there are many penicillin allergies. It's a very common allergic reaction. And then sometimes antibiotics will wipe out your good bacteria and cause superinfections. So superinfections, we already discussed, these happen when antibiotics kill off your good bacteria. They disrupt your normal protective microbes and let the resistant ones take over. So a classic example of this is C. difficile. This can cause very bad side effects like inflammation, diarrhea after the antibiotic is given. So when we wipe out the good bacteria, the bad ones are able to just have like a free pass. And that's why doctors use a narrow spectrum antibiotic if possible. Okay, there are many different categories of antibiotics, and they're broke down based on how they kill or inhibit the microbes. So each class has a unique structure and a unique cellular target, like cell walls or ribosomes. And we already discussed that broad spectrum means they hit a lot of bacterial types, narrow spectrum means they only hit a few. And this is important for doctors when they have to pick the right treatment without wiping out the beneficial microbes. So beta-lactams, these are the oldest and most common antibiotic, and they have a ring structure that lets them block enzymes that build bacterial cell walls. So this causes the bacteria to burst. They're natural penicillins like G and V, and these are good for gram-positive infections, but there are bacteria that has evolved enzymes like beta-lactamases, and these are able to chop open the ring and makes them basically useless. So beta-lactam and its resistance. When bacteria make beta-lactamase, they can literally snip open the antibiotic string, and this is one of the most hard to fight bacteria resistance mechanisms. And to counter them, they will pair the antibiotic with inhibitors. So there's clavulanic acid, which basically protects the drug so it can still work. And a good example of this is clavulanic acid, augmentin amoxicillin plus clavulanate. So not all cell wall antibiotics are beta-lactams. Bacitracin is one of the most common ones. It's used on the skin for like small cuts. But then there's also isoniazid. It's key in tuberculosis therapy because it stops the mycolic acid synthesis. And these are very helpful when penicillins fail to do their job. And so antibiotics, they stop bacteria from making proteins. So protein synthesis inhibitors, these antibiotics stop the bacteria from making proteins, and no proteins means no life. So aminoglycosides, these mess up translation and cause nonsense proteins, whereas tetracyclines, these prevent tRNA from docking, which stops the growth completely. And then there's also drugs that target DNA and RNA. So fluoroquinolones, these stop DNA replication by basically unwinding the enzymes and freezing them, and this stops the copying process. And then rifampin, this targets RNA synthesis and makes it super effective against the Mycobacterium species. And these are powerful drugs, but they do come with side effects and resistance, so they're used very carefully. Membrane targeting drugs. So these drugs work like detergents, and they punch holes in bacterial membranes. So they're strong, but they are risky because our cell membranes are very similar. So it can cause damage to the human and damage our kidneys. Daptomycin, this is used for serious skin or blood infections when other drugs will fail. So bacteria sometimes occur in biofilms like on catheters in hospitals or implants, and these act completely different. So they're able to turn on new genes that make them tougher and less sensitive to antibiotics, making it way more hard to fight and way more resistant. So scientists are trying to find a way to interfere with the quorum sensing, or they have a sticky coating and they're trying to digest that so that the drugs are able to get in. Fungi are tricky because their cells are very similar to ours. So most fungi, antifungals, attack ergosterol, and this is similar to cholesterol. So amphotericin B is super effective, but it's toxic. So azoles are safer and more common for like yeast infections. Now I'm going to talk about the antiprotozoal and antihemolytic drugs. So protozoans are parasites, and they will target the enzymes or the life cycle stages. And quinine used to be the only malaria treatment, but now there's a combination used. This is called ACT. And then there's also antihemolytic drugs. These are used for worm infections, and they're tough because those are parasites. They are animals like us, and instead of targeting reproduction, the drugs will paralyze the worms or cut off their nutrient source so that the body will eventually flush them out. And albendazole, this is a broad spectrum drug, and it's used worldwide in parasite control. So viruses use our cells to replicate, so it's very hard to target them. Some antivirals will stop them from even entering the cells, like Tamiflu that's used for the flu, or others like ATZ. These mimic nucleotides and halt the viral DNA or RNA replication. And then there's protease inhibitors. These stop like viruses like HIV from maturing. So none of these really kill the virus, but it will keep them from spreading until your immune system gets better. Antimicrobial resistance. This means that the microbes can survive doses of a drug that would usually kill it or stop it from growing. So some microbes are naturally resistant, like they're able to produce the antibiotic themselves, but there's also acquired resistance, and this is where bacteria that were once easy to treat suddenly stop responding to the drugs. And this is a growing issue because over time they're becoming more and more resistant and predicted to have more deaths over cancer in about 30 years. There are two main ways that bacteria get their resistance genes. So the first is by random mutation, and this is when DNA will have like a little typo and it'll just help them survive the antibiotics. But then the second is a more common way, and this is through gene sharing. So bacteria can swap resistance through conjugation, transformation, or transduction. And once they get that gene, it will spread throughout the whole bacteria population and make it become more and more resistant. So fungi can also develop resistance in a way that bacteria can't. So they use epimutation, and this is like flipping an off switch. So they basically flip the switch on a gene that the drug would normally target, and when the antibiotic is gone, they're able to flip it back on. And this makes it hard to treat them long term because this mechanism is reversible. So there are 5 ways that bacteria use that they get their resistance, and they can use it in several ways. So they might destroy the antibiotic with enzymes, they might stop it from entering, they might pump it out, change the target so that the drug can't bind, or they can completely rewire their metabolism. And most resistance cases fit into one of these 5 ways. So I'm going to go into a few of them. Enzymatic drug activation. So some bacteria produce enzymes, and they basically are able to chew up the antibiotics. So the best known are beta-lactamases, and they target penicillin and similar drugs by breaking the beta-lactam ring, which is the active part of the molecule, and once that ring is gone, the antibiotic is basically useless. And this is why regular penicillin doesn't really work anymore against Staph aureus strains. So then there is decreased drug permeability. So this is when bacteria basically just closes the door on the drug. So antibiotics need to get inside to work, but if the bacterium mutates the membrane proteins or its channels, the drugs can't enter and can't be used. So gram-negative bacteria, they are especially good at this because they already have that outer membrane barrier. So now I'm going to talk about the metabolic pathway changes. So when antibiotics target metabolism, bacteria sometimes just reroutes the whole pathway. So a good example of this is sulfa drugs. They block folic acid synthesis, but resistant bacteria create a detour using new enzymes. So then there are others that can slow their metabolism or go completely dormant, and these will form persister cells that can survive the antibiotic treatment. And this is especially common in biofilms like those found in the lungs of somebody that has cystic fibrosis. So hospitals, they are basically hotspots for bacteria. Lots of antibiotics, sick patients, and surfaces for microbes to swap out genes with each other. So Staph aureus, I talked about that before, it went from penicillin sensitive, meaning it did respond a little bit, to it's almost 100% resistant now for a few years. And the CDC says that C. difficile is a high threat, and it means that we're running out of options to treat it because it has become so resistant.